Then. Okay, I'll start the class now. So today I will be taking linkage and crossing over. Now, before I start, I'll share my screen. Is this screen visible to you?
Yes, sir.
Okay. So the first thing I'll talk about is linkage and crossing over. This I will take into online and plus offline classes. This particular topic, linkage and crossing over. So you know that in our cells, each of our cells, there are two sets of chromosomes known as homologous chromosomes. One set from which you have inherited from your father, and another set which you have inherited from your mother. And when this crossing over takes place during meiosis, this crossing over takes place between non-sister chromatids. So sister chromatids are these little chromosome lines, these lines of the chromosomes which are green in color, or the ones which are red in color. All the red, this chromatid is a sister chromatid. And even for the green, these two greens, they are the sister chromatids. And this green and red, they are the non-sister chromatids. So the crossing over takes place in homologous chromosomes between non-sister chromatids. As a result, there is exchange of DNA. So part from the red goes to the green and part from the green goes to the red. Because in this crossing over, here is where all the recombination takes place. So initially, we had known about independent assortment from Mendel's crosses. Mendel said that when you cross, say for example, a round yellow pea, which is homozygous, and a wrinkled green pea, which is homozygous, you cross them, you get a round yellow pea itself. And this is a heterozygous. And when you self-cross them, then you get uh, round yellow, wrinkled yellow, round green, wrinkled green in the ratio of 9:3:3:1. So the presence of round in any way did not interfere with the presence of wrinkled. So the yellow can be wrinkled along with round. It can be round. It can be wrinkled and it can be yellow, and it can be green and it can be round. So this is called the independent segregation of two traits, and this is independent assortment. So as per Mendel, Mendel's law of independent assortment states that the alleles in here, yellow or round or more than two of different genes get sorted into gametes independently of one another. So in other words, an allele gamete receives for one gene does not influence the allele received for another gene. So the allele of round did not interfere with the allele for green color, which is another gene. So the seed shape, for the pod shape and the pod color, alleles of pod shape and alleles of pod color, they did not interfere with each other. So yellow did not interfere with round, or green did not interfere with wrinkled. It can be any combinations. But in sweet peas, there was something different found. Normally in pea plant, you always get, whenever you cross these traits in pea plants, you get of F2 offspring, say in the ratio of 9:3:3:1. But in pea, there was something different to say, and that was linkage. So this is then suddenly it was found in pea plants, in sweet pea plants, it was found that linkage between genes, it was discovered in experiment with sweet peas. In sweet pea, there was something different. You did not get a 9:3:3:1. That means one color somehow influenced, one gene, one allele somehow influenced another gene's allele. And therefore, it was called as linkage. So there were deviations from this independent assortment. You did not get in the ratio of 9:3:3:1. So Bateson and Punnett in 1905 did this experiment. So in the traits of of sweet peas, or the characters of sweet pea which is chosen, is that flower color, it can be purple or red. In the previous slide, you had seen purple flower, and here you are seeing red colored flowers. It can be purple or red. Purple is dominant to red, like it is yellow was dominant to green. Here, purple is dominant to red. And pollen shape can be oblong or round. वो लंबा हो सकता है या गोल हो सकता है. It can be long or it can be round. Oblong is dominant to round. So lamba hona, it is dominant to round. So these are the traits which he chose. So he did the cross. Purple long, he took purple long plants which had capital P, capital P, capital L, capital L. Oblong or long, it's the same. And it was crossed with red round, small p, because it is these are the recessive, small p, small p, small l, small l. And as expected, F1 offsprings are purple long, but they are heterozygote, capital P, small p, capital L, small l. So you had these long pollens and purple flowers. It was crossed with red flowers, and you had round pollens. Now the results, you had the F1 offspring, like you know that they were heterozygous, but since purple is dominant over red and long is dominant over round, so you had purple flowers, long pollen, capital P, small p, capital L, small l. So in the F2 crossing, a selfing was done. Capital P, small p, capital L, small l was self-crossed with capital P, small p, capital L, small l. And the gametes, you know, which were formed were capital P, capital L, capital P, small p, capital L, and small p, small l. This you can check in the independent assortment from your class 11 and 12 about Mendel's law of independent assortment. So these F1, they were crossed, self-fertilization was taken place, and you were supposed to have an F2 offspring, purple flowers, long pollen, purple flowers, round pollen. This is the new combination. Red flowers, long pollen, red flowers, round pollen. So you would expect them in the ratio of, the expected number was 240, 80, 80, 27. You would expect them in the ratio of 9:3:3:1. But actually, what was found that instead of 240, there were 296. Instead of 8, there was 19. Instead of 80, you had 27. Instead of 27, you had 85. And the ratio was 15.6:1:1.4:4.5. And this is actually not the 9:3:3:1 ratio. So there was something which has taken place. You did not get the independent assortment numbers. So in the F2 generation, the observed ratio is 15.6 is to 1 is to 1.4 is to 4.5. And the expected ratio 9 is to 3 is to 3 is to 1 is not seen. If you have problems, please do interrupt me and ask questions. So far, okay? Were you able to understand? Hello?
Yes, sir.
Okay. Were you able to understand?
Yes, sir.
Okay. So you see that the parental phenotypes were overrepresented. Who were the parental phenotypes? The parental phenotypes were purple flowers, long pollen, and red flowers, round pollen. They were overrepresented. You had more of these purple flowered long pollens and more of the red flowered round pollens. They were overrepresented. Non-parental phenotypes were underrepresented. That is the new combinations were this 1 and 1.4, they were underrepresented. So this was the evidence that the two genes do not assort independently. The nature of their physical linkage was not apparent to Bateson and Punnett at that time. They did not understand why this was, what was this nature, why were these linked? They did not understand. This is because on these two genes, round pollen and flower color, flower pollen shape and flower color, they were on the same chromosome. So when Mendel did his crosses, the color of yellow or green, yellow or round, they were on different, the pod color and the pod shape, they were on different chromosomes. So आराम से independent assortment took place. But here they were on the same chromosome. So they are physically linked. Therefore, that independent assortment ratio was not seen. In a rubber band or in a thread, if you have two lines, the two lines when you when they are crossed with another rubber band, there is a chance that these two characters they go together. They are not independently assorted. ऑफलाइन बैठ जाओ यहाँ पे बैठ जाओ चेयर लेके बैठ जाओ okay why people are attending so late everything was clear I told everything in the notice board इसके बाद किसी और को नहीं घुसाऊंगा you don't want to do classes don't do it so the linkage is the close association of genes or other DNA sequences on the same chromosomes. The closer two genes are to each other on the chromosomes, the greater the probability that they will be inherited together. So the two genes, जितने भी पास है एक दूसरे के, so chances in the next generation ये दोनों एक साथ ही जाएंगे. फिजिकली एक दूसरे को खींच के रखता है जैसे एक धागा में दो पॉइंट और भी क्लोज रहता है जितना दूर है रहेगा वो धागा उतना दोनों पॉइंट्स उतना इजी है वो ब्रेक करके अलग जगह पे चले जाएंगे लेकिन जितने पास है वो साथ साथ ही जाएंगे इसके मतलब नहीं होता नहीं है होता है लेकिन कम होता है so linkage therefore again linkage is the close association of genes or other DNA sequences on the same. The closer two genes are to each other on the chromosomes, the greater the probability that they will be inherited together. So linkage, recombination, and crossing over, we are trying. So capital L is round, red, long, and small r, small l. So these are two genes on the same chromosomes. So when you cross them, first generation, in the F1, you have this whole chromosome coming, and this one is also coming. Capital R, capital L, capital small r, small l. Meiosis takes place. So you have the first thing which is coming. No crossing over. That is capital R, capital L, and small r, small l. Crossing over capital R, capital L, and here crossing over is taking place. So two things takes place. First, no crossing over. Second, yes, crossing over. So result is the F1 gametes are four types. One gamete which has got the capital R and the capital L. One gamete which has got the small r and the small l. And these are no crossing over. No crossing over has taken place. These are the parental gametes which have come. And the other one where there is crossing over which has taken place, you have capital R, small l, and small r, capital L. So linkage, recombination, and crossing over. Genes that are on the same chromosome travel through meiosis together. However, alleles of chromosomally linked genes can be recombined by crossing over. बहुत बड़ा statement है. Genes that are on the same chromosome, they travel together through meiosis. मतलब जो कि पास में है सेम क्रोमोसोम में, मियोसिस में उन लोग का चांस बहुत ज्यादा है कि एक साथ ही चले जाए. Not the parental ones. That is where there is no crossing over. But alleles of chromosomally linked genes, जो alleles है chromosomally वो लोग linked है, लेकिन when crossing over takes place, ये recombination takes place. ठीक है?
Yes, sir.
क्वेश्चन है? So there are two phases then, coupling phase and the repulsion phase. So a coupling here, this is a coupling heterozygote. Capital R, capital L, small r, small l. And a repulsion heterozygote. So here the parental, this is the parental. So this heterozygote is known as coupling. Same रहता है, जैसे parental में था. But when the crossing over has taken place, capital R, small r, capital L, this is known as a repulsion heterozygote. So now linkage and frequency of recombination. So the same cross again, capital R, capital R, capital L, small r, small l. They are crossed with small r, small l, small r, small l. So red long and white short. This is recessive, homozygous recessive. Normally, this type of a cross is known as test cross, where a heterozygous F1 heterozygous is crossed with a totally recessive for both the characters. It is crossed. It's a test cross. Normally, when you are doing gene mapping, you do use this. And you have in the F2 four types of offspring. One is the coupling phases, capital R, capital L, small r, small l, and this parental, small r, small l, small r, small l, and you have this red short and white long. capital R, small l, and here you have small r, capital L. So this is red short and this is white long.
Long. Yes, sir.
Okay.
Yes, sir.
So what you find is, now out of 1,000 plants, the offsprings which you got, 920 are parental. This was 450 and this is 470. And in the recombinant, it was 42 and 38. You should expect it in 9:3:3:1. You don't get this. Instead, we found that the recombinants are underrepresented and the parentals are overrepresented. So how do you say what is the distance between the two genes? जो जितना पास है, the ones which are the closer together, the more chance is that they will stay together and form parentals. The farther the genes are away from each other, they will the less chance that they will be parentals. They will make recombinants. जो जीन के क्रोमोसोम में जितना पास पास अगर दोनों है तो चांस ज्यादा है कि वो लोग पेरेंटल में जाएगा, और अगर दूर दूर है तो चांस थोड़ा कम हो जाता है वो पेरेंटल में जाएगा. So the frequency of recombinants. So you have 80 recombinants. If you add these two, it is 80 recombinants divided by 1,000. Total 1,000 plants. It is 0.08. Okay, so the gene distance is known as 0.08.
Yes, sir.
So the key points is linkage between genes is detected as a deviation from expectations based on Mendel's principle of independent assortment. And the frequency of recombination measures the intensity of linkage. In the absence of linkage, this frequency is 50%. For very tight linkage, it is close to 0. So, अगर recombination होता है complete, तो 50% chance.
50%
Independent
chance.
assortment होता है, तो 50%. 0.5.
Yes,
और अगर होता नहीं है तो 0.
0.
Yeah, close to 0. This is how you find the map distance. So this is the end of today's class. In the next class, we will talk about chromosome mapping.
Okay, sir.
We will be talking about chromosome mapping in the next class. So this is the basic. Isko go through these lectures once more. Do you have any questions?
No, sir.
No, sir.
No questions. Okay then. Fine. Then I end my class here. Your attendance has already come. Have you joined from your accounts only?
Yes, sir.
Yes, sir.
Okay, then I get the attendance here. It has been tracked. Okay, thank you very much.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
You're welcome.